celebrating 20 years of DNA barcoding. It's 20 years since Dr. Paul Hebert at the University of Guelph in Canada developed the DNA barcoding method for identifying and classifying organisms. Since then, it's become a powerful tool for food safety and food fraud investigations and research. So what is DNA barcoding exactly, and what does it mean for food professionals? DNA barcoding is a method for identifying different species of organisms using a short standardised segment of DNA. It's kind of like scanning a barcode on a product in the supermarket to look up its identity, but instead of a barcode, scientists use a specific part of an organism's DNA. DNA barcoding is useful for identifying species that look very similar or are difficult to classify. For example, if you're trying to identify two different species of fish that look almost identical, you could extract DNA from each fish and compare the DNA barcodes to see if they match or not. In food safety, it can be used for pathogen surveillance, environmental sampling, and outbreak investigations. In food fraud, it's used to confirm the source or species of food ingredients or products, such as by testing seafood samples to see if the labelled species is correct, or if the fish instead has been substituted with a cheaper species. The advantage of DNA barcoding as an analytical method is that you can get reliable information about identity without having to read every single gene in an organism's genome. For example, a human's genome has 7 billion base pairs, and base pairs are components of DNA, but you can distinguish human DNA from all other species by choosing to read just 600 base pairs. Because DNA barcoding methods only look for a small number of standardised DNA markers, which can be amplified and sequenced quite quickly, they can be less expensive than other DNA methods, especially for large-scale surveys. In contrast, whole genome sequencing, which is also used for food safety and food fraud investigations, involves analysing the entire DNA sequence of an organism, which is much more complex and expensive than DNA barcoding. However, whole genome sequencing provides more information beyond species identification, such as genetic variations, virulence, genes, and evolutionary relationships. DNA barcoding in food fraud investigations. Examples of food fraud activities involving DNA barcoding methods include a sausage survey that found 14% of samples contained meat that wasn't on the label, shark fins for soup that turned out to be from protected and threatened species, and mislabelling of fish products with respect to their species at a rate of 15% in Canada.